DNA, or deoxyribonucleic acid, is a molecule found in all living organisms. It carries the genetic instructions used in the growth, development, functioning, and reproduction of all known organisms. DNA is composed of nucleotides, which are monomers that make up the polymer DNA. Each nucleotide consists of three components. A phosphate group forms the backbone of the DNA strand, linking the nucleotides together through phosphodiester bonds. The phosphate group is attached to a deoxyribose sugar molecule, which is a 5-carbon sugar. The deoxyribose sugar is further attached to one of 4 nitrogenous bases: adenine A, thymine T, cytosine C, or guanine G. The DNA molecule consists of 2 strands that coil around each other to form a double helix structure. The strands are held together by hydrogen bonds between complementary nitrogenous bases. Adenine pairs with thymine, AT, and cytosine pairs with guanine, CG. The double helix structure of DNA resembles a twisted ladder, with the sugar-phosphate backbone forming the sides of the ladder and the paired bases forming the rungs. The 2 strands of DNA run in opposite directions, known as antiparallel orientation, which allows for complementary base pairing. The sequence of nucleotide bases along the DNA strand encodes genetic information. Genes are segments of DNA that contain instructions for making proteins, which are essential for the structure and function of cells. Before a cell divides, it must replicate its DNA to ensure that each daughter cell receives a complete set of genetic instructions. DNA replication is a highly accurate process where the DNA unwinds and each strand serves as a template for the synthesis of a new complementary strand. DNA carries the genetic code that determines the sequence of amino acids and proteins. This information is transcribed into RNA through a process called transcription, and then translated into proteins through a process called translation. In eukaryotic cells, DNA is tightly coiled and packaged with proteins called histones to form structures called chromosomes. This packaging allows for the organization and compact storage of DNA within the cell nucleus. DNA is subject to mutations, which are changes in the sequence of nucleotide bases. Mutations can occur spontaneously or be induced by environmental factors, and they contribute to genetic variation among individuals within a species. DNA sequences can change over time through mutations and natural selection, driving evolutionary processes and the diversity of life on Earth. The study of DNA has led to advancements in fields such as biotechnology, medicine, and forensic science. Techniques like DNA sequencing and recombinant DNA technology have revolutionized our understanding of genetics and its applications. This description provides a comprehensive overview of DNA, highlighting its molecular structure, function, and significance in biology.